Okay, so I'm going to then start the meeting. Thank you all for joining in today. As you know, the meeting is being recorded so that we can put it in the website. So today we are delighted. First of all, apologies for the technical problem. There was a problem with WebEx and none of us, not even the speaker, could log in. So we are just a little bit late, but that's all fine. So today we have the pleasure to host Associate Professor Tancredi Caruso, who's now an Associate Professor of Ecology at University College Dublin. His research focuses on the processes that structure soil biodiversity in space and time, and has mostly worked on above-below-ground linkages, which is basically related with plant and soil biota and the ecological networks that these form, and how these networks will respond to environmental changes. So thank you so much for agreeing to be here today, Tancredi. So yeah, the stage is yours.
Okay, thank you, Sonia. I'm going to share my slides, and thank you to the organizer for the opportunity to talk here. Okay, can you see my slides?
Yep, looks good.
Very good, thank you. So again, thank you very much to the organizers for giving me the opportunity to talk about the topic of my talk, which is Barley microbes and their responses to perturbations and environmental gradients. Basically, when it comes to the microbiome of barley, its importance has already been established in various works. This is pretty basic, actually, because we know these days that, in general, the microbes associated to any plant organs are essential to the health of plants at multiple levels, from the physiology of plants to the role of species of plants at entire ecosystem level, especially when it comes to the cycling of nutrients in terrestrial ecosystems. So crops, of course, are no exception to that, and barley, of course, is not. Just confirming that. What is different between crops and plants in the wild is that crops, of course, have been selected to become increasingly more independent from many aspects of the environment, in particular nutrients availability, because in general, in high input agriculture, we provide plants with nutrients. But one of the indirect effects of that has been that plants have become probably increasingly more independent of some of the microbiomes they usually associated with under natural conditions. Now, with the rediscovery of genetic diversity in crops, one of the things we are now discovering is also the genetic diversity of the microbiomes, which is associated with crops. And this is particularly valuable these days because of climate change, or more generally, global change dynamics, more broadly speaking. The role of microbes on plant health and also for crops, it's not just in terms of nutrients acquisition. It's also in terms of a range of other benefits that plants get from their microbiomes. One of the most important ones is one of the most important is the
IBH.
The protection from pathogens. It's been demonstrated for a number of plant species and crops that, for example, are vascular mycorrhizal fungi, which is a major group of endophytes in 90% of plants and also in very many crops. One of the indirect benefits of AMF is protection from pathogens. So, this is well established, and there is already some important amount of publications around the microbiome of barley as well. in wild and domesticated barley as well. And it is known to be important, but it is under-investigated. And especially in relation to how that microbiome can contribute to the ability of crops like barley to respond to an increase, for example, in environmental variability, as we are facing these days with global change, or specific types of stress like flooding stress or heat stress, drought, for example. Today what I'm going to do is I'm going to show some preliminary results that we are getting within a project which I have already had the pleasure to introduce in a previous seminar together with a project coordinator. This is the project RICOBAR, which is a large European consortium in which we are exploring the genetic diversity of barley, very many genotypes of barley around, and how they perform around a broad gradient across Europe. And in that particular project, I specifically coordinate a work package which is concentrating on the rhizosphere microbiome of 6 genotypes of barley that have been selected to represent a gradient of diversity, a genetic diversity in barley. And the results I'm going to present today are basically coming from a pilot we have conducted to start exploring the diversity and complexity of the associations that barley genotypes form with a range of microbes. The overall idea is that anything which is happening above the ground cascades below the ground, and eventually the responses below the ground feed back to the above ground. And the microbes that are associated to the roots of barley are no exception. So any gene expressed by the microbiomes in the roots or in interaction with the roots will have an effect on the overall performance of the plant above ground as well. So we're going to investigate this hypothesis both in terms of a taxonomic description of the microbes associated to the roots of barley. And when I say microbes, I'm specifically referring to both fungi and bacteria. But also another thing we're going to do in the longer term is an analysis, so not just the taxonomic composition of the microbiomes, but also a more functional level description in which we will be achieving by looking to the metatranscriptomes of the rhizosphere microbial community of barley. We have a range of aims in the project, but one of the key aims is it's the characterization of the variation in the associations. Because although there are studies about that, the state of the art is still pretty basic compared to all the knowledge we have on the genetics of barley and also how barley generally performs in a range of environmental conditions. So we know comparatively much less of the rhizosphere than any other above ground trait of barley. And one of the novelties is that we're going to do this at the field scale. So we're going to grow barley in the field in a range of environmental conditions and along this genetic diversity gradient. And we're going to see how the rhizosphere microbiome responds to those sources of variation, either in terms of the genotypes of barley they associate to, but also in terms of how the same genotype can grow different microbiomes depending on the conditions under which it is grown. And as you will see in the next slide, we're going to use 2 landraces, 2 heritage and 2 elite cultivars that have been selected in the broader project. And we're going to grow them across the 4 corners of Europe: north, south, east, and west, under low and standard nitrogen input, just to have this interaction of factors at play between geographical variation, but also management conditions, in this case nitrogen, which is a key focus of the broader project. And then we are going to also have some experiments in the glass house. This is here the range of size that we're going to explore in the experiments. Okay, so this is kind of the long term. So the talk mostly, and I wanted to give you this introduction just to give you a broader context within which the research I'm presenting is pitched. Now, because there is some investigation of rhizosphere microbiome in barley, but it's still not, there's still lots to learn. We have piloted our methods, and we have started an investigation of the variability of the microbiomes of barley in the rhizosphere. And to do that, we have completed a pilot recently in UCD in the field, in which my colleague Sonia has basically conducted a very massive field experiment in which 10 genotypes of barley have been grown under either control or waterlogged conditions. So this was excellent for us because of the incredible number, so the high variety of genotypes we have in this experiment in the field under two extremely different types of conditions in the soil, either control or waterlogged. And we use this experiment to start to explore the diversity of the microbiome in different genotypes, very different genotypes of barley, to see one particular groups of endophytes, the AMF, using microscopic techniques, and to also start characterizing molecularly the microbiomes to see what is the overall variance of the microbiomes and to see how this variance can be affected by control or waterlogged conditions. Now, the first thing to say is that we are focusing on the rhizosphere, and already there, the literature is extremely diverse in terms of what is the definition of a rhizosphere microbial community. Because the rhizosphere hasn't a very strict definition, in the sense that the rhizosphere, or better, you find a number of definitions in the literature about what a rhizosphere is. Qualitatively, holistically, it is the soil which is strictly adhering to roots. That can range from a very few microns to probably a couple of millimeters. What is more complicated is to separate that soil from the roots in a manner which is kind of standardized across your sampling sites. And if you look into the literature, there are very many methodologies that have been proposed to achieve that extraction of rhizosphere soil from the roots of plants, and including crops, but they all revolve around some form of shaking the plants and vortexing the plants more or less delicately until you can, in a somehow standardized manner, extract the soil that is strictly adhering to the surface of the roots. And here I'm just offering you a visual of what that methodology can basically encompass. But the key idea is that we are concentrating on those microbes that are really strictly associated to plant roots, but they are not exclusively associated to roots. So we are not looking to the endophytes, which is the microbes really colonizing the roots, but it's a broader community. It's a community which also is in the soil, but strictly associated to the roots. This means that the type of interactions we're potentially looking at are either direct and indirect. Now, indirect interactions in the microbiomes are very important, actually. It is known, for example, that some of the compounds that roots deposits in the soil may stimulate certain microbes to actually modulate the activities of other microbes. For example, in general, plants will tend to try to fence off any pathogens that can attack the roots. And to do that, one of the things that since plants are capable of doing is to stimulate microbes that are actually competitors of the potential pathogens of plants. So indirect interactions are very important as well, and that's where the rhizosphere is particularly intriguing. Now, in order to have an overview of what microbes are in the roots, we are using molecular methods. We're using amplicon sequencing. I will not go into the nitty-gritty of the details, but basically we are using 16S and ITS as markers of bacteria and fungi. We're using regions that we have piloted and that we are confident are good, are offering us a good cover of the major groups of fungi and bacteria in the roots. One particular group of fungi which is important for crops like barley is the AMF. To do those, we need specific markers. We haven't yet gone into that, so I will not show, I have no data at the moment to show about AMF, but to compensate for that, in the pilot, we have also investigated AMF colonization using classic microscopy techniques, in which you stain the roots and then you look at the microscope at the roots to quantify the degree by which roots are colonized by AMF. And I will show some very intriguing results from that too. And actually, this is coming already now. So you can see here on the y-axis the AMF root colonization percentage, and then on the left and on the right, control and waterlogged samples. And each symbol here, it's a different genotype, one of the 10 that you have seen in the previous slides here. Okay, so each genotype is a symbol in these graphs. Now, as you can see, there's a very clear, neat difference between waterlogged and control samples, and you can see that AMF go up in the waterlogged soil for the same genotypes and barley. In some genotypes, if you look into the symbols, you can see that for some genotypes, that difference is much more remarked than for others. Now, this is very intriguing result, and one of the possible explanations for this result, it remains very speculative at this point in time, but it seems to be validated by other data we have on the barley. One of the possible explanations is that plants under the stress of waterlogging may need to invest in the AMF symbiont because it would help them counteracting the negative effects of flooding. Now, this is not necessarily in terms of better acquisition of nutrients or maybe oxygen. It could also be in terms of protection from pathogens that are notoriously happier when you have wetter conditions in the soil. Things like, for example, fusarium. It is known from other pieces of work that AMF can protect can protect plants from a range of pathogens and plant parasites, including nematodes, for example. And we have actually observed in our molecular data that fusarium goes up in the waterlogged rhizosphere communities. So one possible speculation is that plants are investing more, and some genotypes in particular are investing more in the AMF colonization because perhaps those indirect benefits. But one thing is sure, we have shown here that the great potential for and the flexibility of the same genotypes in terms of how they can recruit more or less an important symbiote as the AMF are. If you look into the number, so the data I'm presenting today in terms of the microbes is mostly the fungi, because this is data we have so far received. We also, as I said before, we're also looking into bacteria, but I'm concentrating on the fungi because we have completed the analysis of those. Here, looking at richness of fungal communities in control and waterlogged, and you can see that although there is some important variation, it seems to be related to the genotypes. In general, the richness of the control plots is clearly higher than that of the waterlogged plots. You can see also by the data points how the distribution of the waterlogged data points is very much, for certain genotypes, very much below the median value. If you look into the actual values here on the y-axis, you can see that the range is massive. We're ranging from above 325 to 200 to below 250, 225 years. So it's it's 100 units of range. So it is pretty substantial as a range. While if you look at diversity indices that account not only for how many species, taxa, I should say, actually ASVs, I should say, amplicant second variants, and their relative abundance, you see much less of a difference. So it's the Shannon index on the y-axis here. Okay, now one of the key words in the title of my presentation is networks. There are, this type of data can be analyzed in very many ways using multivariate techniques, but one of the most interesting ways of analyzing this data is network approaches. Their network approaches are becoming increasingly more popular, and we are going to apply them to this data to explore the structure of the microbiome. In particular, we're going to use what we call network reconstruction models, because there is lots of uncertainty in the reconstruction of those networks of associations, and so we use methods that we have also contributed to have a solid inference of what the structure of the network is. And in particular, the type of networks we are going to look at, they really are in terms of associating certain microbes to certain genotypes. So they, as you will see later, they look like they are what we call bipartite types of networks. So they are in analogy with plant pollinator networks, in which on one layer of the network you have the plants. In our case, we actually have genotypes of barley, and in the other layer of the network you have the microbes at the desired taxonomic level, if you are looking into taxa, or it could be, if you have metatranscriptomic data, it could be different functions that the microbial community is expressing. But before I show the networks, I can show you some basic multivariate analysis of the fungi that already shows that there is a neat difference between the communities of fungi you have in control and waterlogged conditions. So this is a classic coordination in which every point here is a sample, a plot actually, and the symbol, each symbol here is a genotype. And I have just color-coded the samples by either waterlogged or control, and you can see that along the first axis, horizontal axis, all the blue points are on the left, and the yellow ones, the control ones, are on the right. And the reason for that separation is that there are different microbes colonizing the two types of crops. One of the key differences is that certain microbes have been found, only actually fungi, I shall say here, because this is ITS data, have been found exclusively under control conditions, and others have been found exclusively under waterlogged conditions, regardless of the genotype. And then others, the vast majority, have been found both in control and waterlogged conditions, but at different abundances. So in some cases, they will be much more abundant under waterlogged conditions. That is the case of Fusarium, for example. And in other cases, they will be more abundant under control conditions. So already at this level, you can see that there's a clear difference between control and waterlogged. Now, we have a range of information about the plots. For example, we know the position of the plots, and that which it's here described by this variables with this very strange name, PCN-M1, 5, 8, and the y-axis here. Those are variables that describe correlation in the microbial communities over space. So basically, we know that if two plots were closer to each other, they could share some more fungi, regardless of anything else, regardless of whether they were controlled, waterlogged, or whether they were some with a certain genotype or another. But on top of that, you can see the difference in the EMF colonization, and also in the protein content of the barley, which was of the grain, which was higher in the control plots than in the waterlogged ones, and which was anti-correlated to the EMF, which is also interesting. Okay, so this is classic. So now, okay, so what can a network approach add to this? Okay, so we can describe the microbiome taxonomically, and that's a very good thing to do. We can see the correlation between the distribution of certain fungi, for example, in this case, and other aspects of the environment under which the barley was grown. And we can see if there are certain genotypes are preferentially associated with certain microbes or not. So what can a network approach contribute to this? The key idea is that the structure of any ecological network is related to the function that the network contribute to express. So, for example, one of our key hypotheses here is that the structure of the associations between the microbes and the different genotypes, it's an important factor in determining the productivity of the plant community, in this case, our monoculture of barley that you have in the field. So any factor that changes the structure of the microbiome will change the productivity in this case. And then when you release the perturbation, you may have that the microbiome either reassemble towards the original state or just maybe flip into an alternative state. So you compromise the microbiome, perhaps. Okay, that's the key assumption. And the only way to really investigate this is to describe the complexity of the associations using methods that can simplify that complexity. So here I want to illustrate the type of networks we are going to construct, and then I want to show you what we have got for this data set, and then I will conclude my talk. So this is a theoretical example of things you could get. I decided to show that because it's very clear and will help understanding what we actually got. You see here, this black dot here, that represents, it's a node in network language, and they represent, for example, barley genotype 1, say. And I have put here 6 because in our project we're investigating 6 across Europe. And then you see each of these little gray dots here. That is each of those, it's a microbe, okay, microbial species. And the blue lines are what we call the ages or the links. So, for example, all this crown of microbes here have a unique link to this barley genotype 1, which means they've been found only associated to that genotype. While these ones here, they have been found associated only with either these or that. Now you can see that these are, this type of networks can be generated by the real data. We're not reconstructing in the sense that we are using correlations in the distribution of the microbes. We are actually observing a microbe associated to a certain plant. So it's an observed association, okay? And then we use one particular element of this network. You see, for every node, I know exactly how many links that node has. For example, it is 2 for these nodes here, and it is 1 for these nodes here, and it will be much higher than 2 for the nodes in the middle who are connected to all the other genotypes. Okay, and for the node genotypes, they have hundreds of microbes they could be associated to. So one of the things we are trying to do is to see how much we can reconstruct of those networks just by using the information about how many links to each node, because this will give us the potential for this network to rewire regardless of the identity of the microbes. And on the other hand, this is giving us a baseline about the importance of the identity of the nodes in this type of networks. Okay, now this is what our barley networks look like in reality, at least for the data we have so far. When I saw this the first time, the first thing I thought I had to say was coronavirus, because it looks like a bit coronavirus, I have to say. Now you can see here the names here. This is the name of the genotypes we have in our UCD pilot flooding experiment. And then again, each of these is a microbe. So this structure here is what we call a core-periphery structure. So this is very interesting because it means that there is a big number of microbes, in this case fungi, that all genotypes share. That's the core. So there's a core microbiome. At the same time, though, many genotypes, actually all of the genotypes, have some microbes that are uniquely associated to them. So, for example, all of this bundle here of nodes is uniquely associated to Propino, the genotype Propino. And what we have seen, so this is what we will call a module. It's a module because it's from a unique association, okay? now what we observe is that the identity of these nodes and also how similar genotypes are in terms of the node they share, it depends on whether you are under control or waterlogged conditions. Because the genotype is the same, the conclusion is that the plants are recruiting differently depending on the conditions. And so this means that probably there must be a functional reason, some functional reason behind that. Also, because of our network reconstruction approach, we can try to see if these patterns are really due to the identity of the microbes or not, using what we call a null model. And for example, I said that there are modules, and one module is definitely the core microbiome, and another module is, for example, the microbes uniquely associated to a particular genotype. And with this graphics here, you see the histogram here is our null baseline, and the blue line is the observed level of modularity in this case. And you can see that our statistical analysis shows that our observed network are much, much more modular than you were expecting under the random associations of the microbes with the genotypes. So we have proven basically that these associations aren't just random, they really have some structural meaning that we now need to interpret functionally. And the same goes for a range of other network metrics, which I'm not going to go into because they are a bit detailed. But the key message is that the identity of the microbes really matters when it comes to the differences in the microbiomes between control and flooding conditions. And actually, if you look into the details of the analysis, you can see that depending on the conditions, genotypes are more or less similar in terms of the microbe they host. And so now what we're doing now is we are looking to the identity of the microbes to try to have a biological functional interpretation of this. And one of the interpretations we're coming up with is that it's the frequency of the potentially pathogenic microbes, which seems to be going up a lot under waterlogged conditions. In the future, we want to expand this type of analysis to transcriptomics-based data, because there the nodes, rather than being microbial tags, so they can be functions. And some of the functions we're going to explore are things like the ability of nitrogen fixation expressed in the rhizosphere, phosphate solubilization, reduction of oxidative damage, phytopathogen control, which, for example, for the flooding experiment could be very important. And this is work that will be conducted in collaboration with our colleagues in Poland, who are also involved in the broader project. Okay, so that's the end of my talk. I want to acknowledge all the people who have contributed to this, in particular the people in my team, Elia, Shivani, and Nita, and also Sonia, who is here and has provided the samples and the access to the big flooding experiment that I have reported on. Any question would be very welcome, and thank you very much.
Thank you, Tancredi. I start here to see if there's any questions. So far there are no questions here. Is there any questions in the room? I really do not want to ask questions because I'm a little bit involved in this project, so I know a little bit of what's happening. So I'd appreciate.
So Sonia, if
yeah.
no one else asks, I've got a couple of questions.
Yes, go ahead.
Can I?
Yeah, of course.
Thank you for that's a very interesting presentation. I think it's a very timely type of investigation, considering the troubles farmers these days have to sow out potatoes due to flooded fields. My question I have two questions. The first one is related to the differential colonization of AMF as a function of the treatment. I noticed that you have showed us a ratio, so the proportion of root segments that were colonized. I don't know exactly what was in there. I wondered, because you have different genotypes and different conditions, I wonder whether you have a control for the portion of the roots that you are sampling. So paradoxically, could it be that you simply are sampling less roots and therefore you count more mycorrhiza than when they are there? So this, I wonder whether we have you look into it. And that would be the first question. The second question is a technical one related to the very first slide you showed us about the transcriptome of the metatranscriptome of the rhizosphere. So do I understand correctly? So you'll be looking at the transcripts from the microbes in the rhizosphere. And if so, I wonder whether you are considering to map those reads to existing metagenomes to facilitate annotation and then calculation, or you're rather considering to go, I don't know, maybe long reads and then do a de novo assembly effectively of your transcriptome.
Thank you for your questions. They are both not only very relevant to the project, but really, really important questions in general. So the first question, so yes, I mean, the AMF colonization, it's, you know, when we sampled, we had in mind, you know, the type of potential sampling issue you have highlighted. It's very important. So first of all, we look into what the root apparatus of the different plants from different plots will look like. We looked into whether there were major differences between them. Now we were lucky because just under the conditions under which we operate there, the roots are all very superficial, meaning that it's very unlikely we're going to miss the deeper roots. And the other thing is that when you do AMF colonization, you really concentrate on the finest possible roots, ideally hair, roots rich in hairs. And then there is a standardized methods by which you actually sample the roots, like there's a given number of roots fragments that you need to use in your slides. And in the longer run, yes, we will be controlling for the architecture of the roots in the barley. So, you know, clearly, if the conditions are such that a plant is producing a higher biomass of roots, that could itself increase root colonization just by a mere area effect. Okay. But the thing is that what we are observing is that still seems to be controlled by the environmental factor that we are looking at. Okay, so and also the consistency of the results we got between control and model logged let us think that there isn't that type of bias at play in this case for our specific data set. However, it is a fact that quantifying in absolute terms the abundance of endophytes in the roots is a big challenge. So another thing we are trying to do now is we're trying to we want to use quantitative PCR in AMF to really quantify the amount of DNA, of AMF DNA per unit of roots, because that would give us a more reliable estimate of the actual amount of AMF mass material, I don't know how to call it because we're looking at DNA, that you could find in the AMF. And if we find a good correlation between AMF colonization rates and that new quantity that we are looking to measure, perhaps we could better control for the type of biases that you have mentioned. So I think that at the end, it is very difficult to quantify in general fungi and also AMF in roots. But, you know, we have done what other people have done for a long time, and the striking pattern is there. I would be more concerned if I wouldn't see a difference. If I wouldn't see a difference, I would think that there could be that problem at play, because it could be that I don't see the difference in colonization of AMF because there's a response at the root level that is hiding that. But yeah, so yeah, I mean, I'm not particularly concerned in this case, but in general, that point, it's an important point, methodological point to consider, absolutely. The second question you asked, it's about the metatranscriptome. No, we're not thinking of de novo. At the moment, we're mostly thinking of going for known genes and functions, because it's already so many, like because of the amount of genes. But basically, to give you a few figures, at the moment for bacteria, we can annotate about a quarter of the genome. So of all the genes or all the transcripts, 25% we should be able to match to a known function. And some of those functions are particularly relevant to the conditions under which we are stressing the plants, because the method transcriptomics will mostly be applied in two experiments in which we are stressing barley in the glass house. One will be a flooding experiment, which actually Sonia will conduct, and the other one, it's the colleagues in Poland who are actually heat stressing the plants. So because we have specific hypotheses and expectations about the functions that will respond to those stresses, we want to specifically target those known genes. Now, clearly, you're right in that it would be very generally very important to investigate new genes, because we expect plants and their microbiomes to evolve and respond evolutionarily even to things like increasingly more variable climate, increasingly more uncertain environmental regimes, and ideally we want to invest in those crops that are most capable of reacting to that variability, which you could argue is those who are more adaptable in terms of their genomes. And so there might be genes there that we are not particularly aware of that could be particularly important to do that. But at the moment, at least us, we don't have the resources to do that. So we are concentrating on the known genes.
Thank you.
Thanks,
You're
David.
very welcome.
There's a comment here. Is there any specific AMFs that always show together in experiment conditions and not in control? So I think it's just they show only on waterlogging and not in control. That's my understanding. Kurat, do you want to ask this question?
Very good.
Yeah.
We don't know because we haven't sequenced the AMF yet. At the moment, for the AMF, we have some data from the ITS, but the ITS is notoriously not good. We have used ITS2 that offers some cover of AMF, but it tends to represent the most dominant AMF and not the least dominant ones that notoriously aren't well detected by ITS2, which is why when it comes to EMF, we are looking to using more specific markers. What I can tell you, though, is that some of the least abundant AMF tend to be those who make a difference between extreme variation in conditions. So although I cannot answer on the specific data set, I can tell you from other studies, some of which I've been involved in, that there are some relatively not abundant AMF that do tend to differentiate between control and perturb conditions. But I have to disappoint you that I don't have that specific answer for this data set because we haven't simply looked at it yet.
For now. Maybe in a couple of months we might have that answer.
Yeah.
So then we have another question here. How would you distinguish between the root endophytes and rhizosphere microbes? For example, DNA on the surface of roots may be considered as endophytic DNA.
Well, this is a very good point, and it's very true for fungi, because of course, if you have an AMF, it is both inside and outside, because there's an infection inside where the vascules are, where the vesicles are, and then you have the hyphae outside, which is connected to the mycelium and to the common mycorrhizal networks. So of course, if I take roots from the external, microbial biomass, for example, fungal hyphae, from the external of the roots, I could be chopping what's a structure which is actually connecting the external side with the internal part. However, when you study specifically endophytes, what you usually do, and we're doing this in other systems in grasslands, for example, you take the roots, you clean them, you wash them, you sterilize them, and then you extract DNA from inside the roots. This way, you have the endophytes. If there was a fungus which was partially outside and inside, you are still getting the inside bit. If there was a bacterium which was... So the problem is, what is the definition of an endophyte? An endophyte is defined as something that has infected the roots. Some people define endophytes that way, very many actually, I would say. So This means that in order to claim you're looking into endophytes, you are supposed to be extracting DNA from inside the root. And the way people have tried to characterize that has been you sterilize the external surface of the roots, and then you extract the DNA from the inside. The rhizosphere, it's a broader community, and yes, you're correct that it would include some of the endophytes in terms of the fungi, especially. But yeah, in this particular project, we decided to go for the rhizosphere because, in general, you know, the knowledge of the microbiomes of barley, it's not that, you know, there's some good knowledge now, but you know, there's lots to explore. And so we thought that the rhizosphere would be the very first thing to go for. And I think after we have done the study, as other groups investigate the rhizosphere more, probably, you know, we can go more into the endophytes. There are some studies on some particular genotypes on the endophytic community. But yeah, in my view, you first want to have an overall overview of the rhizosphere community, and then you focus and concentrate on the endophytes. So yeah, I hope I have partially answered your question. I could see there was an additional comment as I was speaking, so I don't know.
This is another question. Do you have corresponding microbial community measurements from the soil itself next to the root samples you pulled?
No, we don't have, but this is because it's the pilot. So at the moment we don't. Now consider the growth conditions of this barley. So here, you know, this barley is grown at some high densities. The plants are very close to each other. The space between any two plants, it's little. Arguably, the roots of individual plants are intermingled with each other. So even if you're thinking you're sampling in between plants, you're likely to be sampling lots of roots in those plants. So although we are extracting the rhizosphere soil, you know, so you're correct. Like if I pull out the plant and I shake it and the soil is coming loose, that's going to have a microbial community which is probably different from the strictly rhizosphere one. Absolutely correct. But it is less related to the plant. You could argue you would need to measure both, but if your question is relating genotypic diversity to the environment and to the microbiome, and you have limited resources, the most interesting one to me is the rhizosphere one, because it is the one which is more closely related to both the soil and the endophytic community. It's in between. So, but I agree that for a very complete study, you will need to do the bulk soil. And one of the key reasons for that is that ultimately, plants are recruiting the microbes from the bulk soil. They do have already starting microbiomes in the seed. Okay, so in the seeds, there are microbes already. So if you're sowing the seed and then you have the seedling coming out, you know, there's already some microbes in the seed. But arguably, most of them will be recruited from the soil. So yeah, in a broader, more exhaustive studies, if you sample the bulk soil, possibly even before you put the barley in it, then you have the baseline microbiome that the plant is recruiting from, so to speak.
Then I have, okay, thank you. Then I have another question, and I think I will have to wrap it up soon, right, Malcolm? I was involved in a breeding program for Planet for waterlogging tolerance. Many of the waterlogged roots looked brown and half dead. Do you have pictures of the roots? Okay, so maybe I can answer this.
You should, but there are, yeah, I mean, yeah, sorry, you can better answer, but I can confirm that some of the roots really, yeah.
So basically, in this case, Ian, what we had is that this barley was sowed in April last year, okay, and it was waterlogged in May for very few days. It was waterlogged by 3-4 days. We had very bad weather in Ireland last year, so we had a very, very nasty July, loads of rain. But these barley were sit next to each other, so they were very close to each other on the field. So what we had is that we harvested these plants at the end of the season. So when we were really harvesting the plants, a couple of days before, Tancredi went there with his team and picked up those plants. So they were basically already senescing because the plant has finished all of that, and now what we have noticed, and this is again in controlled conditions in pots, is that we have for sure a very different root architecture. Now I can tell you that we have plans as part of this project that we will look at the root architecture. This will be done with imaging, wind rhizo. Okay, so it's wind rhizo imaging, but we also have, not as part of this project, but myself as part of a different project, a master's student has actually went to these plants and picked up core samples that have been imaged with X-ray CT scanning. So we'll have a complete notion of how those roots look like, but we are still in very early stages. Now, the key thing is that no, they didn't look brown and half dead. They actually look healthy roots, because we didn't collect the roots immediately after the waterlogging. And the plasticity of the roots is very fast from our data. We know that after 1 week of recovery, the plants are basically not having any changes in terms of diameter, volume, etc.
This is very interesting in terms of the microbiome response we observe, because it means that the microbiome is keeping probably the memory of that stress. So the fact, for example, that we observe higher colonization of AMF in the waterlogged ones, this is actually a legacy response to waterlogging,
Exactly. Yeah,
just
exactly.
for the reason Sonia said, which makes the interpretation of this even more intriguing. So the barley was exposed to that stress. Then the stress, so it was a pulse perturbation, okay? So what we observe is the response in the aftermath of that stress. And that is an increase in AMF colonization.
Yeah, there's
so
lots of things to investigate.
Yeah, so that makes it even more intriguing, I think.
Yeah. I think I have one more question and then I'll really finish the questions. So you mentioned that some species of microbes might be associated with waterlogged plants because of increased vulnerability of plants to pathogens in those conditions. This may be speculative, but if you find that certain AMF species are associated with these plants to increase resistance, do you think of inoculating these barley with these AMFs will be beneficial for increasing pathogen resistance in general, or even in non-waterlogged conditions? Or do you think that the interaction of genotype and environment would prevent said AMF inoculation for being applied to barley in general? So we are going into the
Good
realms of
question.
biostimulants, right?
Yeah, I mean, it's a very important question, especially if you consider all the research that is going into the potential for inoculating soils and plants, especially crops, with microbiomes, and AMF is one of the most important groups of microbes that people have been looking into. I think probably my answer would be that at least for the type of data we have, rather than thinking on the microbial side of things, I would go back to the genotypes. I think what we can do with this data is to identify genotypes of barley, in this case, that are better at recruiting the microbiomes that can protect them from the pathogens. So, because actually, if you look into the nitty-gritty of the data we have, some genotypes were better than others in AMF, and maybe less or more associated with pathogens. I haven't shown those data in details, but I think to me the way is more in terms of looking at the microbiome as a trait, and in this case, it's a trait of the barley in the sense that it's like the genotype in the field. It's expressing the trait. I'm using that phrase, but of course, what's happening in nature is much more complex. As you know well, there's this selection process that determines the association between a certain number of microbes and that particular genotype, and this will determine a response of the plant. So to me, it's more like you identify those genotypes that do better in certain conditions. And one of the reasons for that is that they have an association with the microbiome that makes them better, which will be extremely dependent on the match between your genotype and the environment in which you're growing it. And that match is not just the match with environment and the ability of a certain genotype to grow under certain conditions, it's also a match with the genomes of the microbes in the field. Certain genotypes of barley might be more capable of interacting with very common microbes you may have in most soils. Others may need specific microbial associations. So they may be better at certain conditions and certain soils and even by geographical regions than in others. So in terms of application, I think that, I don't know if it is the way in general, but the type of suggestions we may eventually give at the end of our project will be in terms of what genotypes are better given certain conditions. And one of the factors to decide that is the microbiomes.
Mm-hmm. Yeah. And in this specific project that we are talking about, Recobar, we are focusing on waterlogging only in pots. So what Tancredi showed was just a pilot study to look at, to start getting the feeling of the type of results we would get. But the project itself will focus then more on the nitrogen and not as much on the stress. Everything that's going to be examined will be more stressed in controlled conditions. And because it's in controlled conditions, we can actually very carefully wash all the roots and do all the transcriptomics nice and neat, and also do the imaging of the roots as well. Yeah. Okay. I think that's it. Thank you so much, Tancredi, for all of the question, answering all the questions and the presentation. And thank you.
Thank you very much to you, Sonia, and the others, or the other organizers, Malcolm, Ryan, everyone. Thanks, everyone.
Thank you. Bye, everyone. Bye. Thank you. And thank you for the very good questions. Lots of food for thought.
Absolutely. Thank you very much.